RAC is being activated, a cell can become a phagocytic cell. So then around the same time, now all the result was in the fly system, but around the same time, when we found that these results in the fly system, that hyperactivating RAC can make a cell phagocytic, we found there is a paper that came out from NIH where they found that there were human patients with an hyperactive or activating mutation in the RAC, and those patients were immunocompromised and they were immunodeficient and they lacked T cell. And the mutation was a dominant active RAC2 mutation, and that had a lymphopenia, that means a loss of lymphocytes. And the mutation was mapped as to being a RAC2 E62K. So this mutation was changed from E to K at the 62nd amino acid. And then they had made a mouse model of this human patient, and that mouse model also phenocopied the human disease. And you can see in this mouse model, these CD3 positive T cells or CD4 positive T cells, which are helper T cells, or cytotoxic CD8 positive T cells are all low in number, suggesting that these patients have lymphopenia or T cell lymphopenia, or loss of T lymphocytes. So they looked at the thymus of those animals, and the thymus was normal, so there was no problem with the T cell development. And it was a counterintuitive for them as well as for us, because RAC normally promotes the survival and activity of the T cells. So why having hyperactive RAC will lead to the loss of the T cells was the question. So then we found a clue from their paper itself, which is their phagocytes were heavily filled with some kind of substances. So that suggested that actually their phagocytes are doing kind of hyperphagocytosis, because this mutation is not only in the T cells, but also in the phagocytes. And it is most likely that their phagocytes are being hyperactive and they are doing hyperphagocytosis, which is a process of engulfing our substances or pathogens. So we ask the question now that we know in the fly system, if you express hyperactive RAC, you can make even an epithelial cell phagocyte. Then what actually happens, we ask the question, what happens to the patient's phagocytes? Can the patient's phagocyte become hyperactive by this mutation and they're eating their own T cells alive? So to address this question, we got their mouse model, and then we isolated bone marrow-derived macrophages from their mouse femur, but also harvested the spleen and then isolated the T cells. And we did a co-culture of the macrophages with the T cells and did a fixed and time-lapse live imaging to see what happens. And in the co-culture, we have wild-type macrophages with the wild-type T cells. And we also labeled the T cells with a pH-sensitive dye so that once they are engulfed inside of a green macrophage, you can see a yellow signal. And what you're seeing here is occasionally those macrophages in the wild-type macrophages will have some engulfment that is evident by this yellow signal here. But when we co-culture these RAC2 E62K macrophages with the RAC2 E62K T cells, now what we see is much more yellow signal within these macrophages, suggesting, which is also quantified here, suggesting that these macrophages are actually hyperactive and they are eating their own T cells alive. And that is probably a likely cause of why we are seeing a T cell loss or T cell lymphopenia in those patients. So we concluded that the T cell lymphopenia may be due to a combination of macrophage hyperactivity. And I didn't show you the data, but we also saw that this is a two-part system where also T cells are being made hyperactive to engulfment or hypersensitive to engulfment by having this hyperactive RAC mutation. So it's a combination of macrophages being hyperactive so that they can engulf the T cells, kill the T cells, but also the T cells are being more sensitive to engulfment by having this hyperactive mutation. So macrophages can come in a variety of flavors. They could be either pro-inflammatory, where they have an inflammatory signal in response to a pathogen or a danger signal, but they can also be more kind of reparative, where they will be having a more, once you have a wound, they can support a wound healing process. So they will be more anti-inflammatory in that sense. So in the in vitro differentiation space, the so-called pro-inflammatory macrophages are labeled as like M1-like macrophages, which will have a microbial or tumoricidal activity, and they can promote tissue damage. And they have a distinct set of receptor transcription factors, and also they secrete a distinct set of cytokines. as opposed to the more anti-inflammatory ones that have phagocytosis capacity, but also they can help in the tissue regeneration and repair and blood vessel formation and immunomodulation. They have a different set of receptors and also transcription factors and these cytokines that they secrete. So we wanted to see the hyperactive macrophages, whether they are more M1-like or anti-inflammatory or more M2-like. More M1-like pro-inflammatory or more M2-like anti-inflammatory. So again, we harvested the bone marrow, we differentiated them in the macrophages, and then we had a wild type and the RAC mutation macrophages. We did not stimulate them, but in the wild type, we can stimulate them to become M1-like by these interferon gamma and the LPS stimulation, so they become more M1-like. But when we stimulate them with the IL-4, they can become more M2-like. And then we did an RNA sequencing to see how these all four signatures look like. So when we did the RNA-seq, what we found that these, you can see here, the uh, differential gene expression in the RAC E62K, it kind of doesn't fully overlap with the M1 or M2, but it does share a large signature with the M1-like macrophages. As you can see here, there is 1,000 genes which are common between the EK and the M1, as opposed to M2 and the EK. Only 75 are being shared here. So that suggested that the RAC hyperactive macrophages are not totally M1-like, not totally M2-like, but they do share a large signature with this M1-like macrophages, which are anti-tumoral and pro-inflammatory. So then that led us to question, can we, now that we know that these are more anti-tumoral macrophages, can we now make them or design them so that they can kill and engulf the tumor cells? So that's when the CAR-macrophage part of the project came into. So again, the question we asked is, can we harness this hyperactivity of RAC macrophages to treat cancer? And again, I discussed this. That approach that we took is to engineer the chimeric antigen receptor on the surface of this macrophage to target and kill the cancer cells. And the question was again that whether can we make them hyperphagocytic so they can clear the tumors effectively. So using this CAR-macrophage technology, our collaborator, Dr. Megan Morrissey, she had developed an anti-CD19 CAR-macrophage, which is a CD19 positive lymphoma cancer cell CAR-macrophage. So in this case, we have two types of macrophages, one that do not express CAR, control macrophage, the other that express CAR. And CAR macrophages. And the magenta cells here are the lymphoma cells that will express a CD19 antigen. So in the controls which do not express CAR, what we see that they will engage with the macrophages but will not be engulfed. When we have the CAR macrophages, sometimes they can be engulfed, but this process is not very frequent and it's not common in the macrophages. And it was evident that if you can increase this hyperactivity of macrophages so that they can engulf more cancer cells, it will make this therapy better. So then we asked the question, if we hyperactivate RAC in these macrophages, can now we make these CAR macrophages better? And that's what we did when we have a hyperactive RAC together with a CAR macrophages. Now what we will see that these macrophages become hyperphagocytic, hyperactive, and they are now engulfing many more cancer cells as opposed to what we had seen earlier, that the RAC is making them become hungrier and they can engulf many more cancer cells as opposed to the control macrophages, which is also quantified here, suggesting that the enhancement of the macrophages by the RAC can make them a better CAR macrophage. And this is, again, like I said, is dependent on a receptor ligand activation. So if you do not have a CAR, there is no engulfment, not in the wild type, not in the hyperactive RAC macrophages. But when you have a CAR, the wild type macrophages can sometimes engulf one cell, but when you have a hyperactive RAC with the CAR, now they can engulf many more cancer cells. So also more recently, we had published that not only they can engulf cancer cell, but remember the eat me, don't eat me signal that the cancer cells have. So they have a lot many don't eat me signal, and they have a fewer eat me signals. And one of the don't eat me signal is CD47. And we have just recently published that the hyperactive RAC can overcome this problem. So they can overcome this don't eat me signal on the cancer cells. And that is why they can engulf many more cancer cells as opposed to the other regular macrophages that are not able to overcome or bypass this signal. So Again, so that is how we concluded that the RAC enhanced, we call them RAC enhanced CAR macrophage or raised CAR macrophage, that they are able to promote a CAR dependent whole cell engulfment and the tumor killing. So the next question is, can we also, because where the problem is in the solid tumor, can we target the CAR macrophages to kill the solid tumors? So the solid tumor model that we chose to test the raised CAR macrophages is a triple negative breast cancer model. What that means is it's a triple negative. It does not express HER2 or the HER2 as a receptor, or AGF, HER2 or progesterone receptor PR or ER. So it's called triple negative, HER2 negative, PR negative, ER negative. And this is one of the cancers that can actually metastasize to different organs and have a very low 5-year survival. And the only approved therapy is a chemotherapy or PARP inhibitors in these tumors. So right now we wanted to, before going into more in vivo and the patient-centric data, we wanted to see if we can develop a 3D system where we can study this tumor macrophage interaction. And we chose to use this with a collaborator, Pratik Dhawan. We chose to use these chicken embryo chorioallantoic membrane model. What that means is this is a fertilized chicken egg. If we just break the shell and graft some tumor cells, they can form a tumor mass, but they can also pass this membrane and go to the blood vessel and metastasize these organs. So this is a very high throughput, low cost system for us to test if the macrophages will be able to eliminate the tumor cells in a system before going into more in vivo studies. So using these, what we did, we can grow the tumor cells on these chick membranes. You can see that they form these kind of tumor cells, which have also attracted the blood vessels from the chick, suggesting that the tumor cells are using the chick's angiogenesis to harvest their own growth. When we co-culture them with the control macrophages, the tumor does not look any different. But when we co-culture them with the RAC macrophages, now you can see that the tumor mass has significantly shrunk, suggesting that the RAC macrophages can also eliminate possibly or inhibit the growth of these triple negative breast cancers. And then we asked the question, can we now also see that now we have seen that they also recruit blood vessels, whether we can also stop them from harnessing this blood vessel formation or angiogenesis that the cancer cells use to support their growth. So they secrete this VEGF to support their own growth and blood vessel formation. And in these co-culture system, what we had seen is that once you grow these tumors with the RAC macrophages, they do not secrete as much VEGF, suggesting that the RAC macrophages are not only inhibiting their growth, but also inhibiting their pro-angiogenic properties. So suggesting that the RAC macrophages in principle can work with the solid tumors as well. And then since we know that these macrophages, the triple negative breast cancers have only one approved therapy, which is a PARP inhibitor, we try to combine them with a PARP inhibitor. So PARP inhibitors are essentially a drug which are involved in the DNA repair process. What PARP does in a normal cell, in a healthy cell, which is not a BRCA mutant, BRCA is a gene that is involved in the double-strand DNA break repair. If you have a healthy cell, you inhibit PARP. It's a single-strand break repair enzyme. You will have a double-strand break in the healthy cell. But if you have BRCA wild type, that is a double-strand break repair, so it can repair that break, and a healthy cell can survive. But if you have a PARP inhibitor in a cancer cell, then that can lead to a double-strand break. But again, since these are also BRCA mutant cancer, they cannot repair this double-strand break. So they will only kill the cancer cell and spare these healthy cells. So again, a paper was published where they had shown that if you treat the macrophages with olaparib, which is a PARP inhibitor, these macrophages can become bigger and they can also ingest more material. And we see the same thing in the RAC macrophages. So it was rational for us to combine the PARP inhibitor with RAC macrophages and see if they can be more effective in the cancer killing. And the question is again, if we have olaparib or PARP inhibitor, we encapsulate them inside of a nanoparticle, combine them with the macrophages, can we now see a cancer killing? And indeed, this is what we see now. If we have 1,000,000 tumor cells, grow them for 48 hours, and then use 500,000 macrophages for another 48 hours, in combination with the PARP inhibitor, now we see when they have a vehicle, no drug, there's a tumor. When we have a control macrophage with drug, we see some tumor reduction. When we have RAC macrophage with vehicle no drug, we see some more tumor reduction. But when we have both combined, RAC macrophage with olaparib, now we see the tumor is almost killed or gone, suggesting that again the RAC macrophages can already be worked with an approved therapy that is already there for the triple negative breast cancer. So again, suggesting that the olaparib or the PARP inhibitors, which are already approved cancer therapy, can act in combination with the RAC-enhanced macrophages into inhibiting these triple negative breast cancer. So in summary, I have told today that in the lab we had a problem that why hyperactive RAC is killing the egg chamber. We were able to solve that. We were also able to explain why this T-cell lymphopenia happens in the patients and that makes them immunocompromised. But we also were able to engineer a new approach to attack and kill the solid tumor, which is a RAC-enhanced CAR-macrophage. And what we are doing right now is to engineer this using a more patient-centric approach where we harvest the blood from patients, isolate the peripheral blood monocytes, and then differentiate them into macrophages. We express an entire TNBC CAR and the RAC together, and we co-culture them first in our egg model, and then eventually in the xenograft tumor model in the mice, where we can inject tumor cells in the mice that can form a tumor. Then we inject the CAR macrophages, and now we want to see that whether these macrophages can persist, whether they migrate to the tumor, whether they have anti-tumor efficacy, and whether or not they can also recruit a broader adaptive immune response, and whether or not they can also work with the combination therapy of the PARP inhibitors. So with this, I would like to acknowledge our lab, my mentor Denise and Megan, and other people, Mel, Morgan, Anthony, Pratik, who had all been working on this project. And also thank Isha for giving me this opportunity to present the work, some of the work that we've been doing, and I'll be happy to take any questions if there are.